Hello everyone, my name is Pindong Chen. I'm a PhD student in the Institute of Automation in Chinese Academy of Sciences. It's my honor to have a chance to share my study, altered global signal topography in Alzheimer's disease correlates with neurodegeneration pathway. Alzheimer's disease is a typical neurodegeneration disease associated with widespread disruption in different brain areas through disconnection mechanisms. Those widespread disruption breaks the balance between local specialization and global integration in the brain, and consequently impair the daily cognitive ability. The global signal GS refers to the average signal of the gray matter or the whole brain. The GS is suggested to link with the artifacts of head motion, hardware, respiratory, and other unknown effects. Therefore, it was regulated out as a non-neural signal in pre-processing of fMRI. However, there are still some controversies on whether to regulate the GS in fMRI. Recently, some studies suggested that the GS is not merely contain noise. GS could represent an overall fluctuation of the global activity, and the correlation between GS and the local time series could represent a local contribution for GS. The GS were associated with task modulation, weakness, and disorders such as schizophrenia and autism spectrum disorder. But whether the disrupted local global integration in AD impaired the GS and the GSLR is not clear now. Moreover, we should note that the GS is associated with artifacts. How could determine the changes of GS truly reflect the changes of AD is a problem. Fortunately, we have the imaging transcriptomics. Imaging transcriptomics provides a novel perspective to explain the neuroimage. It could recover the underlying mechanism of the alteration in neuroimage based on the spatial association between transcriptomics data and the neuroimage. So, we introduced the imaging transcriptomics to investigate the association between the GIS underlying mechanism and the AD. There is a dataset used in the study. We utilized a large sample of an fMRI dataset from the Multicenter Alzheimer's Disease Initiative, including 257 normal controls, 257 mild cognitive impairments, and 295 AD from Stephen datasets. This is the pipeline of this study. First, the mean time series of gray matter was used as the GS time series, and we calculate the Pearson correlation between GS time series and each voxel time series in the brain. The Pearson correlation map is the global signal correlation map, the GSLR map. We obtain the GSLR map in each group and get the alteration map by group comparison using a general linear model with the gender, age, and dataset as covariables. Then we used the Brainatome atlas to obtain the regional GSLR alteration, and the partial least square PLS regression was applied to investigate the association between GSLR square, GSLR alteration, and the transcriptional data from any human brain atlas. Then we selected the significant genes by permutation, and genes were ordered by the PLS weights. Finally, we applied the enrichment analysis on the significant genes list by the Kyoto Encyclopedia of Genes and Genomes, the KGG. Next is the alteration of GSLR. There is a bidirectional change pattern in AD. The increased area were mainly located in the frontal lobe and the inferior parietal lobule. The decreased area were mainly involved in the temporal lobe and the cingulate cortex. After the FDR correct, the increased areas were mainly involved in the superior frontal gyrus and the middle frontal gyrus. The decreased area were mainly involved in the superior temporal gyrus, lateral occipital cortex, and the hippocampus. Here is the results of the cognitive relevance of GSLR. We extracted the mean GSLR in the decreased area and the increased area. GSLR changes in those regions correlates with the cognitive ability and demonstrate a progressive trend from the normal controls to Alzheimer's disease. The decreased GSLR should difference between groups and positively correlates with the MAC score, and the increased GSLR showed differences between groups and negatively correlation with the MAC score. Next is the results of the imaging transcriptomics. This is the here's component 1 score spatial distribution, which is correlated with the GSLR alteration. And we selected the genes had weight score bigger than 3 and then performed the KGG enrichment. The results of enrichment analysis include several pathways that associate with neurodegeneration disease such as Alzheimer's disease, aging disease, and Parkinson's disease. So in conclusion, our study began with the AD phenotype. We found a bidirectional change pattern in AD, and the change pattern showed correlations with the cognitive ability. Next, we found the genes which is related to the GS spatial alteration patterns. Finally, we found the background molecular mechanism of those genes are associated with Alzheimer's disease. Our study highlights the relationships between local activity and global activity that is changed in AD, and we proved that the informative role of GS in AD. Finally, we provide a new perspective for understanding the functional system changes in AD. In the end, I want to thank my collaborator from the Multicenter Alzheimer's Disease Initiative and my faculty in the Institute of Automation Chinese Academy of Sciences in Beijing University of Posts and Telecommunication and Beihang University. And I also thank the grants that supports our studies. Thank you.